Hello friends, good afternoon and welcome to EduSat Live Lectures. Dear friends, today in the series of genetics, we will be talking about chromosomal mutations. To discuss this topic, we have with us our subject expert, Dr. Charu Dogra Rawat. Dr. Rawat is Assistant Professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you, Amrit. Good afternoon, students. So today we'll be talking about chromosomal mutations. Mutations is any change that occurs in the DNA. This DNA change can be at the genetic level, that is a change or substitution or any kind of damage that occurs at the level of genes, or this change can also be at the level of chromosomes, the entire DNA which condenses to form chromosomes when the cell divides. So today we'll talk about the chromosomal mutations, and particularly the learning objectives for today's session would be to understand various numerical chromosomal mutations involving the full chromosome sets, to understand the occurrence and basis of polyploidy, to understand various numerical chromosomal mutations involving individual chromosomes, and to understand some autosomal aneuploid human disorders. So to start with, what is a chromosomal mutation? A chromosomal mutation, or also called as chromosomal aberration, is a mutation involving a large segment of the genome. So when a larger segment of the or a part of chromosome is basically changed, then it is said to be a chromosomal mutation. Such a mutation usually affects many genes and is often observable at the cytogenetic level. That is, it can be seen with the light microscope. So there are two aspects for a mutation to be called a chromosomal mutation. One, it has to involve a larger segment of the genome and therefore must span a number of genes. And secondly, it must be observable under a light microscope. The chromosomal mutations can again be divided into numerical mutations or structural mutations. Numerical, as the word suggests, is basically the change in the normal chromosome number. It involves a deviation from the normal chromosomal number. And the structural involves the breakage and rearrangements of a chromosome segment. So basically within a chromosome, or one part is basically broken and gets onto a second chromosome, but there is a breakage and rearrangement of the different parts of the chromosome, then it is said to be a structural chromosomal mutation. The numerical chromosomal mutation can involve the entire chromosome sets which is present in the cell. So one set can be 2 sets or 3 sets or 4 sets. So entire chromosomal set is involved, or there can also be basically individual chromosomes that can increase or decrease in number. We will follow this and we will see different examples involving this numerical chromosomal mutations. Structural mutations can again be unbalanced, where the loss basically relates to the some kind of a phenotype or some kind of a change in the function of the cell, or it can be balanced, that the loss is basically compensated by either a gain, or the gain or loss does not affect basically the chromosome number of the cell. So it can be balanced. The balanced chromosomal mutations can be for the single chromosomes, or again it can be of multiple chromosomes which is there. So structural mutations can be the balanced and unbalanced, and balanced can be of single chromosomes or of multiple chromosomes. So we will start with numerical chromosomal mutations that involves full chromosome sets. So before we get deeper into it, let's examine the number of full chromosome sets in a given eukaryotic cell is defined as its ploidy. So what is a normal chromosomal set called as? So this is called as the ploidy. And let's look at some terms used to describe the states of the ploidy, which are, there is a term which is called as diploid. Most of the somatic cells of eukaryotic cells, including humans, is a diploid cell, and diploid means that it contains 2 chromosomal sets. Monoploid term is used when it contains 1 chromosome set. Haploid is used strictly when half the normal number of chromosomes sets are found in the meiotic cells. So when there is a diploid cell, then there is a germ cell development, and when the gametes are formed, they are said to be haploid. Usually they are used synonymously with monoploid, but monoploid basically refers to when there is only a single set of chromosome which is present, and haploid usually is referred to when there is a diploid normal set of chromosomes, or a somatic cell of chromosome is a diploid cell. But when it forms gametes, then usually involved in the sexual reproduction, then there is a halving of the chromosome number, and therefore there is only a single set which is left of the diploid cell, and therefore it is said to be a haploid. Polyploid basically means that more than 2 chromosome sets are present. It is termed as triploid if there are 3 number of sets which are present, tetraploid if there are 4 number of chromosome sets which are present, hexaploid, 6n, etc. Over 10 copies of the genomes are represented by numbers, for example 12 ploid, 13 ploid, etc. So what is the difference between polyploid and polyteny? These are the 2 terms which are often used. So polyploidy, as I just mentioned, refers to the possession of more than 2 sets of homologous chromosomes which are present. But polyteny refers to the possession of many chromatids per chromosome, as in Drosophila salivary glands. So when one of the chromatids keeps on replicating, but it does not get distributed into the daughter nuclei, then basically one chromatid in a particular cell, there are a number of chromatids which are present, and they are all identical. So such a phenomena is termed as polyteny. Then there are other terms which are used, such as isoploid and anisoploid. So isoploid when there is an even number of chromosome sets which are present, and anisoploid when there are odd number of chromosome sets which are present. There are 2 terms, again we will delve deeper into it, which are called as autopolyploid and allopolyploidy. Autopolyploidy is when the polyploidy arises from the intrinsic genome duplication. So polyploidy is basically when there are more than 1 set of chromosomes which are present. So when an individual's genome basically duplicates or triplicates or becomes 4 number of times, then it is said to be a autopolyploidy. Whereas allopolyploidy results from genome duplication of a species hybrid. So if say a species A combines with a species B, and now the hybrid contains the chromosomal sets of both the organisms, then when they duplicate or triplicate, then it is said to be a allopolyploid. When the parent species are known, then it is called as an amphidiploid, basically a tetraploid. When both the parent species are known, then it is also termed as the amphidiploid. We will see that. Then there are other terms which is called as a homoploid and heteroploid. Homoploid is the possessing a chromosome number typical of a species, whereas a heteroploid is basically when the chromosome number set is atypical of the species which is there. So these are general terminology which we use when we refer to the chromosome number sets or ploidy. So let's look deeper into what this polyploidy is, what are the examples of polyploidy, and how does this polyploidy arises. Many multicellular eukaryotes have a diploid vegetative state, which includes the somatic cells, as this represents the minimal ploidy required for sexual reproduction. So you know that in sexual reproduction, the sexually reproducing organisms produce gametes, and these gametes combine to give rise to the next generation. So every time when these gametes are produced, they are produced by meiotic division, which is a reductional division. So this reductional division will cause the chromosome number to reduce to half. So if there was no diploid vegetative state, and only the haploid remains, then at every meiotic cycle, the chromosome number will be reduced to half. So what happens is that a normal vegetative state is diploid. When the gametes are formed, they are haploid. Then 2 different gametes fuse together to again give rise to a diploid germ cell or a zygote, which then develops into an embryo. So all the somatic cells are basically diploid, including the germ cells, but when the gametes are produced, they are haploid. So the minimum requirement of a ploidy for sexual reproduction is a diploid vegetative state which is there. Constitutional changes away from diploidy are poorly tolerated in mammals and birds, leading to grossly abnormal offspring. This reflects the dosage balance of autosomes and active sex chromosomes. What does this basically mean? So, the diploidy is the normal vegetative state, and if it moves away from diploidy, for example, there are 3 sets of chromosomes which are present or 4 sets of chromosomes which are present, then basically it is not tolerated amongst mammals and birds, and it leads to grossly abnormal offsprings, which basically means that the number of autosomes and the number of active sex chromosomes, because the active sex, why we say active sex chromosomes, because if there are 2 homologous sex chromosomes of same type, for example XX in females, human females, then one of them basically becomes heterochromatinized into a bar body to compensate for the double dose of X chromosome which is present. So, there is a ratio of autosome and active sex chromosome which is needed for a normal offspring to be formed. So, now when there are more number of diploid sets which are there, then this correlation basically disrupts, and therefore it leads to a lot of abnormality which is there. Among plants, however, and lower animals such as some insects and leeches, both monopolyploidy and haploidy are well tolerated and is very common in nature. So, polyploidy tend to increase the cell size of the plant. So, in plants naturally it is very common, and in fact plant breeders tend to make polyploidy plant cells. Because they are larger in size, they tend to increase the cell size of the plants, which is a desirable property exhibited by the plant breeders. Economically important, for example, certain examples of the polyploidy plants, economically important triploid plants include several potato species of the genus Solanum, winesap apples, commercial bananas, seedless watermelons, and cultivated tiger lily, which is Linium tigrinum. Diploid bananas contain hard seeds, but the commercial triploid seedless variety has edible seeds. Tetraploid alfalfa, coffee, peanuts, and Macintosh apples are also of economic value. So, a lot of plant breeders tend to induce polyploidy, and this polyploidy varieties are of economic importance. So, they can be of horticultural importance, or they can be of food value increases, but they are very much of economic importance, and therefore plant breeders tend to make these polyploidy varieties. The commercial strawberry, for example, is an octoploid which is there. So, there are 8 number of chromosomal sets which is normally present in a strawberry is present. Now, look at the reason, this autopolyploidy and allopolyploidy. Let us compare that. So, autopolyploidy, as we mentioned, is addition of 1 or more extra sets of chromosomes identical to the normal haploid complement of the same species. So, you can see on the left hand side panel that how these autopolyploid occurs. So, there is a diploid set of chromosomes, and then this duplicates or this triplicates or tetraploidy. So, when they tend to increase in number, there is 1 more or 2 more sets which are chromosomes which are there, then it tends to give rise to a triploid or a tetraploidy thing. Allopolyploidy is for the hybrid. So, when a diploid variety and another species diploid variety, they are crossed, then they give rise to a tetraploid. The F1 generation is a tetraploid containing diploid sets of both the parent species, and then that is a tetraploid which has been generated, but this is a hybrid variety which is there. So, allo is the combination of chromosome sets from different species occurring as a consequence of hybridization. Individual with abnormal ploidy may be viable, but may they also be sterile. Any species which is anisoploid will be sterile due to meiotic failure. So, anisoploid was basically when there was odd number of sets of chromosome. So, even number of sets of chromosome does not pose that much a problem, particularly in plants, etc. When, for example, it is a tetraploid, there are 4 sets of chromosomes. So, at meiosis, the 2 chromosomes can go to one pole, and the 2 sets of chromosomes can come to homologous chromosomes. The different homologous chromosomes can still separate, and both the poles can get 2 of each, and therefore there will not be much of a problem. But when there is an odd number of set of chromosomes, there is an anisoploidy which is there, then due to meiotic failure, one pole or one cell will get two sets of chromosomes, whereas the other will get only one if there is a triploid, if there is a triploidy which is there. So such kind of thing will lead to very much abnormal offspring, or usually it is not tolerated, and therefore it will be a sterile generation which will be there. So now let's look at how this polyploidy arises. This can arise from a cell cycle failure, and what does we mean by that? That it can be done by the omission of DNA replication or unscheduled chromosome segregation to half the ploidy. So for example, there is a normal number of the normal vegetative state is 2n. So during the cell cycle, if the DNA replication does not occur and the distribution into the two daughter nuclei occurs without DNA replication, and then therefore each will get only the ploidy will be reduced to half, and therefore there will be a reduction in the number of ploidy, or there can be an unscheduled chromosome segregation. So when the chromosome segregation has to occur, each pair of homologous chromosomes has to go to different poles, and then that chromosomal segregation is not proper, then again one will receive 2 and the other will receive none, and therefore the ploidy will be reduced to half. Or there can also be the omission of chromosome segregation, that they do not segregate to different poles, or there can be unscheduled DNA replication. Before even the cells are distributed, the DNA is replicated to half, and then therefore due to different failures occurring in the cell cycle, the polyploidy can arise. It can also arise at fertilization. For example, in mammals, the triploidy can result from either diploidy of the sperm or ovum. So as I was telling you that during the the formation of gametes, the haploid gametes need to be formed. But due to any of the reasons, due to cell cycle failure, there can be unusual gametes which are diploid in nature. So if there is a diploid sperm, and this diploid sperm fuses with a haploid ovum, then it will give rise to a zygote which is triploid, because it will contain 3 sets of chromosomes, 2 coming from the sperm and 1 coming from the ovum. So the polyploidy is there. Similarly, the ovum can also sometimes have double the set of chromosomes, and there can be a haploid sperm which fertilizes it, and therefore the zygote will again be a triploid. Or more commonly due to dispermy, where the fertilization of a single haploid ovum occurs by 2 haploid sperms. So these are not normal conditions, but the polyploidy can arise due to any of these mechanisms. The polyploidy can also be induced experimentally, and there have been certain chemicals, or there have been certain shocks, the heat shock or a chemical shock that can be given to a cell, and therefore there might occur a cell cycle failure, and therefore there can be a polyploidy that can be induced. Then there are certain alkaloid which is for example called as colchicine. So colchicine is isolated from a plant, autumn cocum, and from there with this colchicine, when it is when the cells are grown in the presence of colchicine, the chromosomal they are arrested at the metaphase only, so they do not segregate. And when after that it is removed, the cell again enters the interphase stage. So the DNA has replicated, the homologous pairing has occurred, but there is no segregation that is occurring. And because of that, the interphase will again go DNA replication and again presume with the cell cycle, and therefore there will be a polyploidy which is induced. So the potential involvement of colchicine in doubling the chromosome number can be seen here. So if there is a diploid set, and this is an early prophase where there are, the chromosomal pairing is occurring, and in the late prophase when the chromosomal pairing occurs but the segregation does not happen, then when the colchicine is added, they got arrested. Then when the colchicine is removed, the cell again enters the interphase stage, and therefore the cell subsequently re-entering this interphase stage will become a tetraploid rather than a diploid stage which is there. So such chemicals can also induce, and in fact colchicine is experimentally used widespread for inducing polyploidy. We will now look at the origin and propagation of an amphidiploid. So when we were talking about amphidiploid, I told you that amphidiploid is basically an allotetraploid which is there. So there are two different species which when are combined together to give rise to a F1 generation which is a polyploid generation, then it is termed as an allopolyploid. And when the parent species are known, then usually it is termed as an amphidiploid. So how can this amphidiploid be seen? For example, there is a species 1 of genotype AA containing 6 chromosomes A1, A2, A3, 3 chromosomes and homologous pairs A1, A1, A2, A3, and there is a species 2 which is capital BB genotype and containing 2 chromosomes B1 and B2. So when they form haploid gametes, because it was a diploid state, then the gamete will be A1, A2, A3 and B1, B2, single A and single B of the genotype. So they will be haploid. When the fertilization occurs, you will get a hybrid. This hybrid will contain A1, A2, A3, B1, B2, which is basically half the set of the original chromosome which is there, and therefore it will be a sterile hybrid. But if we induce chromosomal doubling at this stage, for example, the usage of colchicine, etc., then the chromosome will double and it will give rise to a tetraploid, allotetraploid called as the amphidiploid, which will be completely fertile. Because now it contains a complete set of chromosomes coming from species 1 and a complete representation of the chromosomes coming from species 2, and they can give rise to a diploid gametes which are there. So they will give rise to fertile amphidiploids which are there. So that is how the amphidiploids are formed, or allotetraploids are formed, and they are propagated. So species 1 contain genome A consisting of 3 distinct chromosomes and 2 contain genomes of B consisting of 2 distinct chromosomes which are there. Now, a classical example of amphidiploidy in plants is the cultivated species of American cotton, Gossypium. The species has 26 pair of chromosomes, 13 are large and 13 are much smaller. Old world cotton had only 13 pairs of large chromosomes. In fact, that instigated into the scientists working on Gossypium that there might be a allopolyploidy which is occurring in there. And when they looked at the wild American cotton, they revealed there were 13 pairs of small chromosomes. So actually the cross between the old world cotton and the wild American cotton would have given rise to this hybrid which is called as the Gossypium. J.O. Beasley reconstructed the origin of cultivated cotton experimentally by crossing the old world strain with the wild American strain and then treating it with the hybrid with the colchicine to induce the diploidy, to double the chromosome number, and therefore they could get the fertile allotetraploids or amphidiploids which were completely fertile and hybrid variety which was there. Some other examples of amphidiploid if we look at is the hybrid formed between the radish, Raphanus sativus and the cabbage Brassica oleracea. So if you look at this, the Raphanus radish, the diploid state is 18 chromosome, and in fact the Brassica cabbage, the number of chromosomes is again 9 in a haploid set. So if both of them were crossed together, they gave rise to a hybrid which is called as a Raphanobrassica or rabbage, where the 2n is equal to 18, but it is a sterile variety because there is not a diploid representation of each species which is there. Now, if this diploid rabbage is treated with colchicine, it will induce the doubling of the chromosome, and therefore a tetraploid or 4n rabbage will be obtained. So this is the again a typical example of an amphidiploid, a generation of an amphidiploid which is there. But unfortunate with this wonder plant was that it had the leaves of radish and the roots of the cabbage. If it would have been vice versa, it would have been of economic importance, otherwise it wasn't of any economic importance. The successful commercial hybridization uses the grass wheat and the rye. So there is a bread wheat, Triticum aestivium, and there is a rye, and then when they are crossed together, they give rise to another variety which is called as triticale. So wheat and rye are crossed together to produce the hybrid triticale. Now the bread wheat, basically the chromosome number is 7, and the triploid gamete, their 3n is equal to 21, and they can occur as a tetraploid or hexaploid sporophytes. So if this tetraploid sporophyte, for example Durum wheat in the lower panel, is crossed with the rye, The non-natural occurring rye or the cultivated rye is basically diploid saprophyte and sporophyte, and diploid sporophyte, the chromosome number is 14. So, if the rye and the durum wheat, for example, they are crossed, they will give rise to the triticale species, which will be basically 6N in number, or a hexaploid, which is equal to 42. So, typically they will combine together, they will give rise to a sterile hybrid, and if you treat them with a colchicine, then it will give rise to a fertile amphidiploid, which will be there, or fertile species which will be there. And this is basically of very much economic importance, because as you can see, then it has much more grain and it has both the properties of the rye as well as the wheat which is there. There is another term which is used, which is called as endopolyploidy. So, endopolyploidy is the condition in which only certain cells in an otherwise diploid organism are polyploid. So, not the entire, all the cells are not polyploid, but only some of the cells are polyploidy. In such cells, the set of chromosome replicates repeatedly without nuclear division. So, a typical polyploid would be that when the chromosome sets are more in number, but when some particular chromosomes replicates frequently, but they are not distributed without the nuclear division, then it is said to be a endopolyploid which is there. The example is in vertebrate liver cell nuclei, including the human ones. They often contain 4N, 8N or sometimes 16N chromosome sets. Stem and parenchymal tissue of apical region of flowering plants, they are also often endopolyploid. The cells lining the gut of mosquito larvae, they attain a 16N ploidy. The reason for endopolyploidy is not very clear, but basically maybe the proliferation of chromosome copies often occurs in cells where high levels of certain gene products are required. We will see this as a part of numerical chromosomal mutations, where basically a part of the chromosomes duplicates. We will look at that, and then we will look at the gene amplification part, where they assume that why certain chromosomes are replicated, because they might the cell might be needing the high level of their gene product which is required. It is well established that certain genes whose product is in high demand in every cell exist naturally in multiple copies in the genome. For example, ribosomal and transfer RNA genes are the example of multiple copy genes. So, such chromosomes are replicated, but they are not distributed. So, this is a phenomenon of gene redundancy and gene amplification which is there, and it leads to what is called as the endopolyploidy. So, we will look at the numerical chromosomal mutations when individual chromosomes are involved. Again, before we looked at different examples, we will look at the terms used to describe the number of individual chromosomes in a karyotype. A karyotype is basically a shorthand representation of the number of chromosome sets and sex chromosome configuration of a particular cell. For example, a human chromosome, for example, a human cell karyotype is 46 plus XX or 46 plus XY for a normal male karyotype, and a karyogram or ideogram is a pictorial representation of all the chromosomes where the homologous chromosomes, where they are paired together and then distributed into different groups. We will take up this karyotype in detail in a different session, but right now we will just focus on the mutations which can be represented, which can be observed in the karyotypes of different disorders which are there. So, some of the terms we will quickly go through are the euploidy and aneuploidy. Euploidy is possessing the full chromosome sets having extra or missing chromosomes. The chromosome imbalance, the loss or gain of chromosomes with consequent multiple gene dosage effects. Hyperpolyploidy or hypopolyploidy. Hyperpolyploidy is basically possessing more chromosomes than usual, and hypopolyploidy is possessing fewer chromosomes than usual. Pseudodiploidy, possessing the correct diploid chromosome number but an abnormal karyotype due to simultaneous monosomy and trisomy for different chromosomes. And this monosomy and trisomy basically comes into aneuploidy. The eusomy is the correct number of copies of a given chromosome. So, we are not talking about the complete chromosome set here. We are talking about the individual chromosomes. So eusomy is the correct number of given copies of a chromosome which are present, and aneuploidy is possessing the incorrect number of copies of a given chromosome. For example, nullisomy, when there is no copy of a particular chromosome which is there, monosomy, when there is only one copy which is present, disomy, when there are two copies which are present, polysomy, when there are more than two copies of a particular chromosome which are present, and it is called as trisomy when there are three copies, tetrasomy when there are four copies, and so on. So we will discuss about these numerical chromosome mutations involving individual chromosomes in our next session. Thank you. So in this session, we will talk about numerical chromosomal mutations that are involved in the individual chromosomes. There are different terms which are there which we talked about. So euploidy and aneuploidy. I'll continue with this aneuploidy, which is the extra or the missing chromosomes which are there. So aneuploidy is the deleterious in both plants and animals, and the effects are more severe in the animals which are there. The loss or gain of chromosomes. So aneuploidy is basically that the individual chromosomes are either lost or they are gained. So this loss or gain of chromosomes causes chromosomal imbalance with consequent multiple gene dosage effects. The genes which were present on one of the chromosomes, when they are lost, then their function will be lost and therefore it will have an effect. So there will be multiple gene dosage effects because multiple genes will be lost and there will be a chromosomal imbalance which will be there. In humans, such effects are lethal with 22 possible autosomal trisomies. Only there are 3 which are normally found in human live births, and only 1 is viable in long term. So autosomal trisomies or autosomal aneuploidy, they are not tolerated in humans. So we will look at certain disorders which are there, which are arising due to this autosomal trisomies which are there before going on for the sexual differences in the chromosomes or sexual aneuploidy due to the sex chromosomes which are found in the humans. So one is a trisomy 21 called as the Down syndrome. So as you can see, this is a karyogram which is a pictorial representation, as I explained, of the chromosomes of a individual. So this is a 46 chromosomes plus XX chromosomes which are there. So in trisomy 21, if you can see the 21 number chromosomes shown by an arrow, they are 3 in number. So this is a Down syndrome. It occurs in 1 in 700 live births, and the clinical phenotype includes mental and growth retardation, characteristic facial features such as upward slanting eyes, small nose, open mouth and protruding tongue, low set ears, partial syndactyly and a simian crease which is usually there, lower life expectancy than normal, and many die in the childhood. So in the photograph, a female suffering with Down syndrome can be seen on the left hand side with a normal sister which is there. So this occurs because there is an extra set of extra chromosome number 21 which is present in the human. Then there is trisomy 18 which is called as the Edwards syndrome, and you can see the arrow is pointing towards where there are 3 chromosome number 18 which is present. It occurs in 3 to 6,000 births which are there. The clinical phenotype includes elongated head with low set dysplastic ears, curved feet, clenched overlapping fingers, and cardiovascular and renal disorders, and there is always a perinatal death which is there. Then there is a trisomy 13 which is called as the Patau syndrome. So there are 13 number chromosomes which are present. This is a male karyotype which is shown. The occurrence is 1 in 5 to 10,000 live births, and the clinical phenotype includes gross head and brain malformations as can be seen in the photographs, the reduced or missing eyes. Polydactyly, there are more number of fingers and toes which are there, the low set and dysplastic ears, cleft lip and palate, fused nostrils and cardiovascular disorders, and again there is a perinatal death. So out of the 22 autosomal trisomies which can occur, these are the 3 trisomies which only leads to the live birth which are there. The numerical chromosome mutations, when we talk about sex chromosomal aneuploidy, this is better tolerated because of the dosage compensation mechanism which act to redress the natural variation in sex chromosome number. So we already know, for example, in human females there are 2 X chromosomes which are present, and in human males there is a single X chromosome. Its constitution is X and Y. So because the female contains 2 X chromosomes, it will contain double the dose of genes which are present on X chromosome. So now to create a genic balance, the one of the X chromosomes gets heterochromatinized into a bar body, and this is called as a dosage compensation, because if the females would have double the dose of X chromosomes, then there will be a lot of difference between a female and a male. So to make the X chromosome genes in single dose in both the individual of the same species, one of the X chromosome basically heterochromatinizes and becomes inactivated, and therefore there is only a single. This is called as a dosage compensation mechanism. Now this dosage compensation mechanism is already taking care of the natural variation in sex chromosome number which is existing, and therefore the sex chromosome aneuploidy, as we will see in the human disorders, it's basically better tolerated. One of the disorder is called as the monosomy X or a Turner syndrome, where in a female one of the X chromosome is not present. So you can see that there in the karyotype, in the karyogram, that there is only one X chromosome which is there. It occurs in 1 is to 10,000 live female births, and the clinical phenotype as shown in the photographs includes short stature, webbed neck, wide carrying angle which is cubitus valgus, widely spaced nipples, primary amenorrhea, and infertility which is there. So this is the condition when there is an XO of the condition which is arising. There is a condition called as trisomy X or a triple X syndrome. So in this particular case, in the female there are 3 X chromosomes which are present. So there is a mutation which leads to an extra X chromosome in a particular female which is there. It occurs in 1 is to 1000 live female births, and the clinical phenotype may include failure of the development of primary and secondary sexual characteristics and mild mental retardation. Usually the individuals having trisomy X are normal and fertile. Another one is the polysomy X with Y, termed as the Klinefelter syndrome. So there is a 47 plus X and Y is the karyotype. So there are 2 X chromosomes which are present, primarily associated with the karyotype 47XX and Y. So in this case again one of the X chromosome will become a bar body, but still there are 2 X chromosomes which are there. It occurs in 1 is to 1000 live male births. Clinical phenotype may include infertility, breast development in males, testicular atrophy, malformation of the sex organs, non-functional sperm, tall stature, and mild mental retardation. The increase in severity, there is an increase in number of chromosomes. So with the increase in number of chromosomes, there is increase in severity, and there can occur 48 and triple XY or 49, 4 X's and Y, Klinefelter syndrome conditions can be there. So, more the number of X chromosomes, then more the severity would be there. So, again this is mutation due to the presence of 2 extra or 3 extra chromosomes in a X chromosomes in a male which is there. Another one is called as a disomy Y, where the Y, the male depicting chromosome is present in 2 in number as shown in the karogram. This is called as the super male syndrome. It is primarily associated with the karyotype 47XYY. It occurs 1:1000 live male births. Clinical phenotype includes tall stature. The karyotype is tenuously associated with increased tendency to antisocial and violent behavior, although there is no conclusive evidences to show this, and individuals are usually fertile. So, basically the clinical phenotype is that they are a little taller in their stature, and but this does exist, the super male syndrome does exist where the chromosome number is increased. So, sex chromosomes, they are comparatively still tolerated, but autosomy, trisomy or anything is completely intolerated in autosomes, and only 3 live births can be seen which are there. So, aneuploidy, what are the reasons for this occurrence of this aneuploidy or increase or decrease in the individual chromosome number? They are caused because of 2 reasons. One is non-conjunction and another is non-disjunction. Non-conjunction is when the homologous chromosomes fail to pair during meiosis. So, when they fail to pair, then there is a wrong distribution of the chromosomes that occur, and therefore it gives rise to these trisomy or nullisomy and all these kind of conditions which are there. Another thing is called as non-disjunction, when the chromosomes do pair but the segregation is a problem. The non-disjunction can be primary non-disjunction when the homologous chromosomes pair fail to segregate at the first meiotic division, or when the homologous chromosomes pair fail to segregate at the second meiotic division. So, you can see that on the left hand side panel there is a first division which is their non-disjunction has occurred and 2 chromosomes have gone to one cell, and the other cell has a nullisomy condition where there are no pair of homologous chromosomes, or not a single representation of the homologous chromosome is there. Then in the second meiotic division, basically which is basically mitosis, and when they are distributed into 4 cells, then such condition arises that 2 of them will contain 2 pairs each and 2 of them will contain no representation of that particular chromosome, and when then this fertilizes with a haploid gamete, this will give rise to a trisomic or a monosomic condition as shown. If this non-disjunction occurs at the second meiotic division, which is the mitotic division, if you can see, then the first one is a normal disjunction and both the cells have got single chromosome, and then when the single chromatids divides in the gametes, then there are gametes which are containing only 1 chromosome and 1 chromatid, and in some containing 2, and in some again there is no representation of that particular chromosome which is there. So, this will give rise to, or this will when combined to a haploid gamete will give rise to a disomic and disomic conditions which are normal conditions, but still it will be accompanied by a trisomic or a monosomic condition that can occur. So, aneuploidy can, the main reason for the occurrence of this aneuploid gametes or with an abnormal number of chromosomes is basically because of the non-disjunction of a particular homologous pair of chromosomes that occur during the meiosis. Another type of mutations are termed as the structural chromosomal mutations. So, we talked about the numerical chromosome mutations where the number of the chromosomal sets or number of the individual chromosomes were differing and giving rise to the altered phenotype. The chromosome, within one chromosome there can be alterations, and these are termed as the structural chromosomal mutations. They are caused by the faulty repair of the chromosome breaks or by recombination between homologous but non-allelic sites. So, when there is a rearrangement or when there is a breakage in an individual chromosome, then it is said to be a structural chromosomal mutation. What are these breakpoints if we look at? The point at which the fracture or recombination occurs is termed a chromosomal breakpoint, and structurally rearranged chromosomes are termed as chromosomal derivatives. So, the point at which this fracture or recombination is occurring, where this faulty repair or where this recombination between homologous but non-allelic site is occurring is said to be a chromosomal breakpoint, and the chromosomal, it is called as a chromosomal derivative. Now these structural mutations can be categorized into balanced or unbalanced one. So balanced when they do not, when there is a loss and simultaneously gain of a chromosome, for example. So the number of chromosome will not change, then it is said to be a balanced chromosomal mutation. There can be unbalanced when there is actually a complete loss of one chromosome, or there is a complete insertion of a chromosome, or there is insertion of one single chromosome set, because now we are not talking about individual chromosomes loss and gain, we are talking about parts of the chromosome. So one part of the chromosome is completely lost and the genes present on that segment is also completely lost. Then there will be an unbalanced in the dosage of that particular genes which are there, and therefore this will be called as the structural mutations. The animal and plants differ in their tolerance of structural mutations. The animal gametes tend to be functional even if severely unbalanced and will take part in fertilization, normally generating an unbalanced zygote. So they will still be fertile and they will be fertilized and they will give rise to an unbalanced zygote. So generally it tends to be there. The same is true for plant ovules, but not for the pollen grains. So if the pollen grains, they have structural mutations or aberrations which are there, they tend to abort and the fertilization does not take place. We will look at the unbalanced structural mutations first. They involve the loss or gain of genetic material and behave as partial monosomies or trisomies. So monosomies was when there was a complete loss of one particular chromosome which was there. So there is a homologous pair of chromosome which is present in a diploid cell, and if, for example, X chromosome was completely lost in XO, Turner syndrome, it was said to be a monosomy condition. Or there is a trisomic condition when the entire chromosome is present extra, for example, trisomy 21 or trisomy 18. This unbalanced structural mutations is actually partial monosomy or partial trisomy, because one segment of a particular chromosome will either be completely lost or will be an extra present because it will be duplicated or insertion which will basically be there. The structural mutations involving DNA loss are termed as the deletions or deficiencies. For example, there is a chromosome which is shown and probably it contains segments which are A, B, C, D, E, F and G, and there can be a deletion of a particular segment D, and that is termed as a structural mutation. The structural mutations involving DNA gain are termed as insertions or duplications. For example, the normal is A, B, C, D, E, F, G, and now there is a duplication of a segment BC. So there are two segments which are present in one single chromosome, that is said to be a duplication or an insertion of the BC segment which is there. And these are on unbalanced structural mutation because either there is a loss in the genetic material or there is a gain in the genetic material which is occurring. Deletion can occur now either near one end, which is termed as terminal deletion. For example, you can see that the break point is near A and B, and therefore the part A is lost, or it can also be within the interior of the chromosome, and such deletion is called as an intercalary deletion. For example, you can see the intercalary deletion, the entire portion DC is lost. So it forms a kind of a loop, and this loop is completely lost. As you can see, there is a formation of a deletion loop which is there. So when the homologous pairing occurs between a deleted chromosome, deleted region C and D chromosome, and a normal chromosome, then there is a loop that tends to be formed, and that is called as a deletion loop which is there. So it can be in the terminal deletion or it can be an intercalary deletion which is there. Even the smallest visible deletions affect several megabases of DNA and therefore span many gene loci. They often cause what are called as contiguous gene syndromes, that is, disease phenotypes resulting from the simultaneous loss of many linked genes which are there. For example, some human syndromes are given here. For example, the Cry-Du-Chat syndrome. The karyotype is the deletion, the fifth chromosome on the short arm. You can see the karyotype of the Cry-Du-Chat syndrome here. Then in the fifth chromosome where the red arrow is shown, there is a deletion of it is a terminal deletion. There is a terminal deletion of the in the P arm of the chromosome which is there. And the clinical symptoms is basically it's a small head with associated mental retardation and characteristic cat-like cry. There are other syndromes, for example, WAGR syndrome, where there is a deletion of the 11th chromosome, and the longer arm, which is called as the P arm, the 13th number segment is basically deleted. And the clinical symptoms include Wilms tumor, emerydia, ambiguous genitalia, and mental retardation, which leads to the, it's an acronym. The clinical symptoms, WAGR is an acronym for all these clinical symptoms which are characteristic for this particular disorder. Then there is a Prader-Willi syndrome which is there, in which there is a deletion on the chromosome number 15 from region Q11. So, short arm 11 to 13 portion is lost from the paternal chromosome which is there. And the clinical phenotypes, there is a small stature, obesity, small genitalia, behavioral difficulties, and mental retardation. So, you can see because of the deletion of certain segments of chromosomes, basically the different disorders are occurring. In fact, in Cry-Du-Chat syndrome in 2004, it was reported that the portion of the chromosome that is missing contains a particular gene which is called as the TRT gene. This encodes the telomerase reverse transcriptase, which is an enzyme essential for the maintenance of telomeres during DNA replication, and therefore it was not able to maintain its terminal or end, and therefore it was shortened and it has lost that part which is the P terminal part which is there, leading to the syndrome called as the Cry-Du-Chat syndrome which is there. The duplications, the effect of the duplications of a particular gene segment, it may result into gene redundancy. This has clearly been the case with the gene encoding ribosomal RNAs or the rDNA, which is needed in abundance in the ribosome of all cells to support protein synthesis. For example, in the common intestinal bacterium E. coli, about 0.7% of the haploid genome consists of rDNA, the equivalent of 7 copies of the gene. So, there is basically the segment of a particular gene which are more in number. So, there is the rDNA, for example, the ribosomal genes, because the cell needs a lot of protein synthesis, therefore, the gene redundancy is there. More number of that particular gene is present. In Drosophila melanogaster, 0.3% of the haploid genome is equivalent to 130 gene copies consist of this ribosomal DNA. The second way in which the amount of rRNA, for example, is increased is termed as the gene amplification, where the replication of a particular gene occurs without its nuclear division. So, in the frog Xenopus, for example, the nucleolar organizer region NOR contains the equivalent of 400 redundant gene copies coding for rRNA. To further amplify the number of rRNA genes, the rDNA is selectively replicated and each new set of gene is released from its template. Because each new copy is equivalent to one NOR, multiple small nucleoli are formed in the oocyte. So, basically these duplications, where they occur naturally, and these duplications lead to the gene redundancy and the gene amplification, which is required by the cell for increasing, for example, the ribosomal RNA copies required for the protein synthesis by these cells. The duplications may also cause the phenotypic variations, and one example is the bar mutation in Drosophila. In the early 1920s, Alfred H. Sturtevant and Thomas H. Morgan discovered and investigated this bar mutation. The normal wild type Drosophila females are basically B plus and B plus, having about 800 facets in each eye. So, you can see the different phenotypes of different Drosophila females. The heterozygous females, which are B and B plus, they have about 350 facets, so they are reduced in number. While homozygous females on an average has only about 70 facets. So, you can see that homozygous females on the right hand side, the eye is reduced and disc shaped. It is basically the disc shaped which is there. The females were occasionally recovered with even fewer facets and were designated as double bar with the genotype of BD and B plus. And this was observed because of this particular reason. So, wild type, there was a region present on this particular chromosome which are single in number. So, the bar region gave rise to a normal wild type phenotype. In the heterozygous bar, there is a duplication of this bar region in one of the chromosomes and will lead to the heterozygous bar phenotype. The homozygous bar will contain this duplicated region on both the homologous chromosomes, and therefore the disc facets will further reduce. And the double barred region in one of the homologous chromosomes, you can see that the bar region is present in triplicate, and in one of the homologous chromosomes it is only present in single. So there is a duplication of a particular region which is leading to the phenotypic variation, as can be seen in the eyes of these particular Drosophila which are there. About 10 years later, Calvin Bridges and Muller compared the polytene X chromosome banding pattern of the bar fly with that of the wild type fly. And if you see this comparison, it was revealed that one copy of the region designated as 16A, as shown in the diagram, is present on both X chromosome of wild type flies, but that this region was duplicated in bar flies and triplicated in double bar flies. So this particular region, which is shown in red here, is the 16A region, and this 16A region was either duplicated, as can be seen in the heterozygous bar and in the homozygous bar flies, and even triplicated, as can be seen in the heterozygous double bar phenotype which was observed. So duplication did lead to the phenotypic variations which was basically there. There is a simultaneous whole arm deletion and duplication that can occur. So that was the examples which we were talking about is leading to an unbalanced structural mutations which are there. When there is a deletion of a particular segment, for example in Cry-Duchat syndrome, or when there is a duplication of a particular segment, for example this bar region in the Drosophila which is there. Sometimes there is a whole arm deletion and duplication that can occur when the division of the centromere is at the wrong place, and it generate chromosomes with two long arms and no short arms, and vice versa. So you can see there is a homologous pair of chromosomes which are there, and normally there is a vertical division which occurs, so that the short arm and the long arm of one chromosome remains with one and the other chromosomes remain with other. But sometimes an abnormal horizontal division can occur, leading to the short arm only present in one of the chromosomes and longer arm only present in the other set of the chromosome, and then when it gets duplicated, then basically this is there. Such perfect metacentric chromosomes are termed as isochromosomes, and they cause monosomy for one arm and trisomy for the other, with corresponding gene dosage effects. So because of this abnormal horizontal division and duplication, you can see that the isochromosomes have arisen. The shorter one only has the small arms and they do not have the longer arms which is there, and therefore it will be monosomy for one arm and usually the trisomy for the other that occurs, and therefore the genes present on the one which is lost will be lost, or there will be more number of genes which are, when they are trisomy, they are present. So there will be a corresponding gene dosage effects which are there. So we discussed about the structural chromosomal mutations, and in structural chromosomal mutations we have talked about the chromosomal rearrangements which are basically unbalanced in nature. So deletions and insertions. So in our further sessions we will talk about the balanced structural mutations that occur, such as inversions or such as translocations, and some other mutations such as the occurrence of the fragile sites which are there, we will continue to do in our next session.
I would like to thank ma'am for this very interesting discussion, and then I would like to thank you dear friends for watching our show. On that note, dear friends, we will bid you goodbye. Thank you for watching our show. Stay tuned.